four factors that affect enzyme. So we have enzyme concentration, substrate concentration, temperature, and pH. So these are the four factors that you need to know how they affect the rate of an enzyme catalyzed reaction. Let's start with enzyme concentration. And so if you look at this graph, so we have concentration on the x-axis and rate of reaction on the y-axis. So you can see that as we increase the concentration, the rate of the reaction is increasing. All right, so why is that happening? Let us look at two containers. If you have container A and container B. So remember now, with this graph, your substrate concentration must stay constant, but we're increasing the concentration of the enzyme. So let us say we have 3 substrate molecules. So these are the substrates in the container. All right, 3 substrates in each of them. So the substrate concentration, it must stay constant. Now the enzyme concentration, I'm going to put 2 enzymes in this container. All right, for the rate of our reaction to increase, it means that the enzyme must be colliding with the substrate. They cannot just collide, but when they collide, they have to do so in the correct orientation. That means if this substrate hits this enzyme in this fashion, a reaction will not occur. Remember, it only takes place when the substrate binds to the active site. In the container, the enzyme and the substrate will be colliding, but no reaction will occur unless you get an enzyme substrate complex. And to get the enzyme substrate complex, it has to bind to the active site. So it's not just about colliding, it's about colliding in the correct orientation. So if you have 3 substrates and 2 enzymes, and in this one, you have 3 substrates. Let me put the next enzyme. In which of these 2 containers, A or B, which one do you think more collisions will take place in. You can press the answer if you don't want to speak aloud. So this container A, it has 3 substrate, 2 enzyme. All right, so everyone is saying B, right? That is correct. So if more collisions are occurring in B, which container will have faster rate? Is it A or B? Which one will have a faster rate? B, exactly. So I am showing you this diagram that you'll see that is the reason why as you increase the enzyme concentration, the rate of the reaction will increase. Because once you increase collision, what you're doing is increasing the chances of getting an enzyme substrate complex. So the quicker you get your enzyme substrate complex, the faster the rate will be, because it's the enzyme substrate complex that will lead to the formation of the products. All right. The more enzyme substrate complex you can form, the faster the rate of reaction. So as you increase enzyme concentration, the amount of collisions that are taking place is going to increase. All right. And so the rate will increase. Also, if you look at this, right, you only have 2 enzymes here. You have more enzyme than, you have more substrate than enzyme. That means when these 2 substrate bind to these 2 enzymes, this one will actually have to wait until one of these reactions has been completed for it to be converted into product. All right, so for that reason, when you increase concentration, the rate of reaction is going to increase. I'm going to clear the screen and put it in words. So I will allow you to take off the graph, and if you want to take the diagram as well. All right, so I'll give you a minute to do that, and then I'm going to clear the screen. All right, I'm going to clear the screen now. Let's erase these. So we are doing enzyme concentration. So there are 2 things that you can be asked, right? You can just be asked to state the effect of enzyme concentration and rate. So if you're asked to state the effect, you do not offer any explanation. All you need to say, as enzyme concentration increases, the rate of the enzyme catalyzed reaction increases. So notice I did not offer any explanation because all they said was to state the effect. So the effect is as the concentration increases, the rate of the reaction also increases. Now, if they ask you to explain now, that is where we tell what is actually happening. Your graph, this will only occur if the number of substrate is high. If you get a graph that's showing enzyme concentration and you see it is tapering off, that means substrate concentration is low. So it is limiting the rate. So if you get a graph where you see its rate versus enzyme concentration, and you get a straight line, and you see a curve or broken line, but there's a flat line. Where you have the flat line, the substrate concentration So when this substrate concentration is high, the enzymes, as you increase the concentration, the rate will be going up. But if you see that the rate is getting constant, it means that the amount of substrate has decreased. So it is limiting how fast the reaction can actually occur. On a rate versus enzyme concentration graph, it is typically a straight line. But if you get one where you have the straight line and you see it tails off, this part here means that the substrate concentration is low. So it is limiting the overall rate of the reaction. All right. So that is it for enzyme concentration. All right, I'm going to clear the board now. So if you don't finish writing, just take a picture or a screenshot. All right, so again, let us go back to our containers. All right, so what we're doing is, we're looking at the effect of substrate concentration. Now, with this one, the substrate concentration, the enzyme concentration is going to stay constant, and we're going to increase the substrate concentration from one to the next. All right, so this is container A, this is container B, this is container. I'm going to do 3 containers. All right. All right, let us just say the circles are the substrate. All right, and I'm just going to put some triangles for the enzyme. So remember, its substrate concentration must stay constant. And then we increase the enzyme concentration from one container to the next. All right, so remember, this is the enzyme, and these are the substrate. So question, in which of these containers do you think the rate is going to be the fastest? A, B, or C? C. Right, because as you know, by now, the more particles present, what we have to be careful is not just the more particles, but in this one, the more enzymes you have present, the faster the rate is going to be. So if you look at container A, you only have 2 enzymes. So again, collision has to take place, not just collision in the correct orientation. So as you increase the concentration of the enzyme, you are increasing the chances of getting a correct collision, meaning one that leads to the formation of an enzyme substrate complex. So when you increase the concentration of the enzyme, you are increasing the chances of getting an enzyme substrate complex. So as you increase concentration, collision increase, and that increases your chances of getting an enzyme substrate complex. The quicker you get the complex, the faster the rate will be. So when you look at this, this has two enzymes that is going to be shorter. Well, it will take a longer time than when you have four enzymes to collide. And in this, you have six. So you know by now, when you increase concentration, you literally see that you're increasing the amount of enzyme, the same amount of substrate. Collision increase, more enzyme substrate complex formed, rate increases. All right. If you want these diagrams, just take a screenshot. I am going to erase it and put it in words. All right. Someone asks if B and C would have the same rate. It would not have the same rate for a reason being, remember now, I see why you might ask that, because you see 4 and you see 6 enzymes. So you're assuming that when you get 4 enzyme substrate complex here, you would get 4 here as well. And so these 2 enzymes would not be in use. I don't know. I was saying that someone asked if B and C wouldn't have the same rate. So the reason, I'm assuming that the reason why they asked, right? If you have 4 enzymes here, you would get 4 enzyme substrate complex. And you have 6 enzymes down here, but you only have 4 substrate. So you would still get 4 enzyme substrate complex. While that is correct, rate is how quickly you get them. So not because both of them are forming 4 enzyme substrate complex, which one is going to do it quicker? Because remember, these reactions would have to be timed. So they are experiments. So let us say we are looking for a color change then, all right? And we're timing them for 1 minute, all right? So this one will not give you 4 enzyme substrate complex in 1 minute. Let us say this one give you 1, this one give you 2, and this one give you 3 in 1 minute. So that is the importance of the rate. You are timing it for a specific period, all right? Eventually, all of them will form 4, but in a given period, the one with the more enzyme, it would form it quicker. Okay, so we are not looking at the amount, we are looking at how quickly it forms it. Not that both of them can give 4, but how quickly it does. All right, so I hope that cleared it up for you. Why this one would not have the same as this. All right, I'm going to clear this now and put it in words. All right. Also, just look to the right of your screen. Here, where you have the flat line. When you see this here, it means that all of the enzyme sites are used up. All right, so so for this one, basically, this is occupied with this. So these 4 are occupied. All right. And there are no more substrate to be catalyzed. So remember, in the enzyme concentration 1, when you add the horizontal line, it's because the substrate was limiting the rate of reaction. In this one, all of the enzymes active sites are occupied, so there is nothing else to do. So the rate will flatline, basically. All right. Remember, you see these lines here, the vertical or plant line, it means that the reaction is still going on. You have more occupied enzymes, so the rate will increase. But once you get here, the enzymes active sites are occupied. All right. All right, so let me put that in words now and move on to the next two factors. All right, so just like before, they can ask you to state the effect. Wait a minute. Sorry, just realized. View the other way around. Hold on. Good thing me pick up. If we're doing substrate concentration, wait, it is the enzyme that has to be constant. So for substrate concentration, we want to see how substrate affect it. From one container, we have to increase the amount of substrate and keep the enzyme constant. All right, so just switch it around where the circles are the sub. Oh, that's how I have it. Wait. So sorry about that. When we're doing substrate concentration, the substrate must increase and the enzyme it must stay constant. All right. So this should have been the enzyme. So the enzymes stay constant throughout, but we increase the substrate. The explanation is this same, You have more substrate, collision increase, you get more enzyme substrate complex. But it's just that for this, you keep the enzyme constant, you keep the substrate, you keep increasing the substrate. All right. So for this flat line up here, it would be in container C, where you have 4 enzymes, but you have more substrate. But the rate cannot go any further because all the active sites are used up. All right, so that was the correction. You keep the enzyme constant and you increase substrate. All right. All right, so state the effect. All right, so this is your explanation over here in black. This here in blue is just explaining the horizontal part, why the rate is constant, if it does. All right, and let's say they ask you a question, right? So they give you the graph and ask you, what does this line means? All right, well, you would know that the rate is constant. If they ask you what adjustments you could do to increase the rate again, like that, then here in red is the answer. You would have to increase the enzyme concentration. So once the rate gets constant, if you want to increase it again, you would increase the enzyme concentration. That is when you're looking at the effect of substrate. If you were looking at the effect of enzyme, then when it gets constant, if you want to increase it again, you would increase the rate of the, sorry, you would increase the concentration of your substrate. All right, so that's it for the effect of concentration of substrate and enzyme. We're going to do pH and temperature. All right, so I'll give you a minute, right? And then we'll move to pH.
Sir, I have a question.
Sure.
In this case, would substrate concentration be the limiting factor or the enzyme concentration?
The enzyme.
Okay, sir.
Right. So whichever one you're looking at, if you're looking at substrate concentration, enzyme is going to limit it. Right, and we can look, so as once you use up all of the enzymes, then there will be no further increase. So if you keep increasing the substrate, but the enzymes stay constant, will reach a point where the enzymes will become saturated. Mhm. Right, I'm going to share the board now. All right, so the graph for pH and temperature will have a bell-shaped curve. So it's not like the enzyme. It's not like the enzyme and the. It's not like enzyme or substrate. All right. So now it's effect of pH. All right. So All right, when it comes to pH now and temperature, enzymes have a particular pH at which they work best. What is that? So for the pH, where the enzymes work best, what is that pH called? Anybody?
The optimal pH.
That is correct. And so on this graph, the optimum pH is where I would get my maximum rate, which would be here. I would identify this peak and draw a line to the y-axis. And whatever number is there, that would be my maximum rate. My optimum pH would be 7. Maximum rate would be here. So Vmax for my maximum rate, it would be the peak here. All right, so how does pH affect enzyme? When you come to pH now, you have to remember that enzymes are protein. And they are held together by certain forces. All right. So let's say this is your protein. Let me see. All right, so it's folded. Let's say all right, I need to do it bigger. All right, so let's say this is a protein and it's folded, right? The reason why this can fold there, let's say you have an amino acid side chain here, right? NH3. The NH3, it will have a positive charge. And let's say down here, you have a particular side chain, which is COO minus. All right. This positive charge and this negative charge will attract each other. All right. So there will be an attraction. So that is what is holding this part here to this. So the reason why it curves like this is because of the bonding between the amino acid side chains from different parts of the polypeptide. So look, so when I did protein, you learned about primary, secondary, and tertiary structure. The primary structure would be the long chain of your polypeptide. It hasn't been folded yet. All right. So this now can be folded by interaction. So let me put NH3 here. All right, positive. And let me put the COO minus here. All right. So this is able to fold by the interaction between amino acids side chains. All right. Because I want you to understand what is actually happening. This is the structure of your amino acid, the general structure. It have a carboxylic group, C-double-bond-O-OH. So this is your acidic group, all right? And this is your basic group, NH2, all right? So when it is in a basic solution, this group here, it can gain when it is in an acidic solution, this basic group here, even if you don't do chemistry, just know that when you put an amino acid, not an amino acid only, when you put a protein, acidic solutions, this group here on your protein, it is going to pick up a proton, which is hydrogen, all right? If you put this in acid, this is going to pick up a hydrogen and it is going to gain a positive charge, all right? So let us say before you put your acid or change the pH, this group, it was NH2, all right? If you put it in an acidic solution, it is going to gain a charge, all right? Let me just show you something else up here. Let us say this was NH2, right? And there was no interaction between these two. because you get the interaction if this is negative and this is positive. All right. Remember now, once you change the pH, how the enzyme is folded, it is going to change. And the reason for that, when you change the pH, the bonds are going to be adjusted. Let me explain now. Here, you have NH2. Give me a second. You have NH2, it does not have a charge. So this is representing one amino acid, right? Up here. This is the next one. Let us say you increase the pH of the solution. So it is a basic solution. In a basic solution, sorry, keep on saying that. Let's say you decrease the pH, right? It's an acidic solution. This group, it will gain a hydrogen. When it gains a hydrogen, it will gain a positive charge. Now remember, these two did not have an interaction. But if you decrease the pH, means it gets acidic, all right? This is going to pick up a hydrogen. And so it becomes NH3. Once that happens, this is going to gain a positive charge. Now, these two groups that were not interacting before can interact, all right, and form a bond. Good. So you have just changed something about the enzyme because these two were not bonding before. Now that they are bonding, the shape is going to change. You understand? Once you adjust the pH, these groups will be adjusted. And once they adjust, the bonding is going to change. And so the shape of your protein is going to change with the enzyme. All right. Now, let us say this is how we started out. Good. And you adjusted the pH by increasing it. So you make it become basic. This group here, this acidic group, it can pick up a hydrogen. If it gains a hydrogen, the negative charge is going to disappear because it is picking up a hydrogen. So there's no, this interaction between negative and positive, that's the ionic interaction. So if this gains a hydrogen, this interaction no longer exists. There's no longer an ionic interaction. So before it was positive and negative, and so you have that interaction. But now there's no negative charge, so there's no interaction again. This shape will change. All right? Is that clear? Any feedback? You can text if you have any question. No. All of this explanation is just to let you understand it for yourself. I will put it in words what you need to know. All right? So when I say that, when you change the pH of the solution, bonds in the enzyme are going to break, and the enzyme changes shape. This is the result. When I say bonds are going to break, that is what actually happens. So it is the side groups of your amino acids that will change. All right. So this might become NH3 or NH2, depending on what you do. This might become COO minus or COOH, depending on the changes in pH. But just know that once you change the pH, these charged groups, something will happen. And once it changes, the bonding is going to change. And so the overall shape of it is going to change. The changes in pH is affecting the amino acids. Remember, your active site, it contains amino acids as well. All right. So pH, amino acids, their side chains are being changed. All right. And that causes the shape to change. All right. So if you don't have any question, I'm going to put it in words now. Give me a second. All right, let me erase this. All right, if you want to draw this, just take a picture. I'm going to erase it now. So if you want it, take a picture. Give me a second to plug in my laptop. All right, I'm ready again. So in terms of this graph here, I'm going to state the effect like this. So as a pH, so what I'm doing is stating the effect. Somebody asking a question? Yes, yes, you can say that.
Oh no, let's leave this.
Right. Let me just add, well, it's there already. DNH, when the enzyme, or let me point out on the graph, this is the optimum, right? So the enzyme is working at its peak. This here, the enzyme is not denatured. Here, the enzyme is not denatured. It's still working. But enzymes, they have a range of pH at which they can work. So the only time the enzyme is actually denatured is if you go far from the optimum. So for example, this enzyme, its optimum pH is 7. That means from here, can mark it here to about here. It's still good, all right? From here, it's almost basically denatured. Once you are getting a rate, it's still working. So it doesn't mean that once you see it starts, once you mean you pass the optimum, it is denatured. All right, that is not the case. Once you pass the optimum, it is not denatured. It is great. So denaturation, you can look at it as a gradual process where the enzyme, it starts to change shape. So the more you change the pH, the enzyme starts to change shape. So when you make a significant change now, that is when it completely loses its shape. So if it is not a significant change in pH from the optimum, your enzyme is not going to be denatured. Its shape will change enough. So don't mix up the changing of the shape with denaturing. So once you change the pH, the enzyme, its shape will change. And that is why the rate will change. And for a pH graph, the best way to view it in terms of understanding it, start from up here. So start from pH 7, all right? pH 7 is the maximum. When you go to pH 6, you're moving away from the optimum. When you go to pH 8, you're moving away from the optimum. So if you view it from the optimum pH, you realize that going away from the optimum in any direction is causing the pH to, sorry, it is causing the rate to decrease. So going away from the optimum pH in either direction causes the rate to decrease. But even though when we look at the graph, we say that as pH increase, rate increase, that is true in a sense. But if you look at it from this perspective, from the optimum, you will realize that deviating from the optimum pH, the rate decreases. And that is what we put in the explanation. So once you change the pH, the bonding in the enzyme is going to change. So the rate automatically changes. It doesn't matter whether you increase it or decrease it. Any form of change in pH changes the enzyme, the rate is going to change. The significant change now, that is what will cause it to be denatured. All right, so if it is not significant, it will not be denatured. It will just start work slower. All right, so that is it for pH. The final one we have to look at is temperature. So I'll give you a minute or so, and then I start again. All right, I'm going to erase now. That is what I'm going to try to do by 2:30. So we're going to do the temperature, and then the inhibition part not is not too long. Alright. But I will do it even if we end at 2:40 or so. I am going to do it. All right, so for temperature now, this one is a bit easier, and we are familiar with it from CXE. All right, so temperature. So for temperature and pH, it's the bell-shaped curve. All right. So as we know, this temperature here, all right, so I can put 0, 50, let's say 25. So this would be our optimum temperature. We will get our maximum rate at this point. All right, so that's Vmax. So this point here, that's Vmax, our maximum rate. All right. All right, so state the effect. That's the V is for velocity. So it's the maximum velocity or the maximum rate. I'm continuing it over here. All right, so that is what temperature. So the first half of the graph, let's look at it. This half of the graph over here, that is for this part where we speak about temperature increasing, collision increases and rate increasing, right? So this part is the first half of the graph. First half of the graph. That's the explanation for over here. And then this part is for the second half of the graph. All right, so in the first half, no bonds are going to break. It's just about increasing collision and enzyme substrate complex, so the rate increase. Once you get to this point now and beyond, that is where the bonds start to break until the enzyme is eventually denatured. Right, I'll give you a next minute and then I'll clear the board.